to say go. Today, I'm going to talk about using brain imaging and biomarkers for diagnosis and investigation of Alzheimer's disease, especially about the Alzheimer's Disease Neuroimaging Initiative. For those of you who are not that familiar with Alzheimer's disease, it's associated with two proteins. The first is amyloid beta, and the second is tau. And the disease leads to memory dysfunction, memory decline, ultimately producing dementia. Until recently, the Alzheimer's disease has been defined clinically with autopsy confirmation. And it was not possible to definitively diagnose the disease until we had autopsy. But today, with some of the biomarkers that I'm going to talk about, it's possible to determine whether somebody has Alzheimer's disease in the brain quite definitively in someone who's alive. Currently, there are no effective treatments for Alzheimer's disease. Some treatments are being developed and clinical trials are underway. Some of the treatments are aimed at amyloid protein. I'm going to point to this just to get used to using the pointer. Some are aimed at amyloid. Some are aimed at tau. Some treatments are aimed at neuroprotection. A decade ago, there was a lot of chaos in the field of Alzheimer's disease biomarkers, and as various companies and laboratories were developing potential treatments, there was a need for optimization and standardization of biomarkers for clinical trials. So this led to the Alzheimer's Disease Neuroimaging Initiative, which has been a very large project. We've received over $140 million so far from the National Institutes of Aging and other institutes of NIH, which are listed. And we also received about 30% of our funding from our industry partners. In addition to me, I'm the principal investigator. You can see that there are lots of other people who are involved with this project whose names are listed here. And I'm going to, in the next few minutes, give you a lot more descriptions of what's going on with this project. The Alzheimer's Disease Neuroimaging Initiative, otherwise called ADNI, was funded in 2004. It's continuing today and will run at least another 2.5 years, and we're planning a 5-year renewal after that. So hopefully it will be going at least 8 or 9 more years. We've enrolled, as you can see, trying to get used to using this clicker. 350 normal subjects, 150 subjects with subjective memory complaints, 850 subjects with mild cognitive impairment, 350 subjects with dementia due to Alzheimer's disease. So overall, you can see it's well over 1,000 subjects have been enrolled, and they're all followed longitudinally at 57 sites across the United States and Canada. You can see on the map the location of the various sites. Everybody gets a clinical evaluation, blood tests, lumbar puncture, various cognitive tests to measure memory function and other kinds of brain functions, various types of MRI, which I will describe, 2 kinds of PET scans, a fluorodeoxyglucose PET scan, which measures brain metabolism, and an amyloid PET scan, which measures brain amyloid. Lumbar punctures are done for measuring the A-beta and tau proteins in the cerebrospinal fluid, and we're getting genetics on everybody, and I'll tell you a little more about that. One thing that makes this project quite unique is that all of the data from ADNI is in a public database, and you can see the URL here. The URL is usc/loney/ADNI, and anybody can go to this database, get a username and password, sign the data use agreement, and anybody can get all of the data, all of the raw data, the images, the clinical data, the cognitive data, all of the genetics data. It's all available to any of you, anybody you know who wants to download the data and write papers. There's no embargo on the data, which means that if a patient were scanned today, for example, in about 2 weeks, the MRI scan would be up on that public database. So there's many, many thousands of scans of all types and data that's linked to the scans, and it's all quite well organized. So we encourage people to use the data and write papers, because that's That's the point of the project. So this next slide shows pictures of a lot of the types of MRI scans that are done. You can see we do T1 weighted scans for brain structure. We do T2 weighted scans, FLAIR to measure white matter hyperintensities in the brain, diffusion tensor imaging, and then on the bottom row, arterial spin label perfusion MRI, resting state task-free functional MRI, fluorodeoxyglucose PET scans, and amyloid PET scans. All of these scans are done on almost all of the subjects, although the DTI scans, the ASL MRI scans, and the fMRI scans are vendor specific. That is, the DTI scans are acquired on the Philips scanners, and the ASL scans are acquired on Siemens scanners, and the fMRI scans are acquired on the GE scanners, whereas all of the other MRIs, the T1, T2, and FLAIR, are acquired on all scanners. And then everybody gets FDG PET, and everybody gets amyloid PET. This is a picture of the hippocampal subfield. So we do a particular high-resolution scan to parcellate the hippocampal subfields. This is a separate project funded by the Alzheimer's Association. And you can see from this little slide that says manual labeling how you can draw the CA indentate gyrus and the CA1 field, the subiculum, the entorhinal cortex, and so forth. And again, all of this data is available on the LONI database. So this next slide shows some of the amyloid scans that we've been getting, some examples. And on the top is PIB, Pittsburgh Compound B, which was the first PET scan using carbon-11, which was able to detect amyloid in the living human brain. And below that is Florbetapir, which is now widely and commercially available. And the top two rows are on a normal healthy control. So you can see there's kind of a bluish or greenish color there in the brain, whereas the lower two rows are PIB scans and Florbetapir scans on a patient with Alzheimer's disease. You can see there's a lot of red there, especially in the outer parts of the brain where the gray matter is, showing that amyloid protein was detected there in abundance in this patient. Also, on the far left, you'll see these numbers 0.94 below PIB and 1.01 under Florbetapir, and then 2.17 and 2.01. And those numbers refer to the total uptake throughout the whole brain, reference to a region in the back of the brain. And we tend to use these numbers for a measure of the total brain amyloid. So this next slide is a very important slide because it is a histogram of the distribution of these amyloid PET scans with Florbetapir. This data is now a year old. We have quite a bit more data now. Unfortunately, I'm having trouble getting this thing to click, and let me try something again. I'm unable to point to the columns here, so I'm just going to have to describe it. The normal people across the top of this, you see a histogram that says normal N263. You can see that the bars are highest to the far left of that graph, where it says Florbetapir negative. And you can see a horizontal line that tends to separate that cluster of subjects from clusters which are more to the right, which are Florbetapir positive. So what that means is that normal people are largely amyloid negative. They don't have a lot of amyloid in their brain. These are people about average age about 73. But there are quite a few people that are to the right of the line. That is, they have amyloid in the brain, which means that they have Alzheimer's disease. They have plaques, and most likely they have tangles. But these are normal people. So we call that preclinical Alzheimer's disease. That is, they are people who are healthy, they don't have a problem, but they have amyloid pathology in their brain. And as we'll see, they're at greater risk for some problems in the future. If you go to the bottom, where it says AD on the right, N of 146, you'll see that most of those people are to the right of that vertical line, and not too many of them are to the left of the vertical line. So that means that most patients with Alzheimer's disease clinically have a lot of amyloid in their brain, but it also points out that a fair number of people diagnosed with Alzheimer's disease clinically don't seem to have amyloid in their brain, and we think that means they probably don't have amyloid pathology. They probably don't have Alzheimer's disease, even though it was diagnosed clinically. If you then look at the late MCI and early MCI populations, you can see that they have a big distribution. Some of them, they have people that have a lot of amyloid, but there's also bars on the left, especially in the early MCI, where there's a lot of those people who are amyloid negative. So MCI doesn't necessarily mean that you have early Alzheimer's disease, but about 60% of patients with MCI seem to have early Alzheimer's disease. And we'll see more about that in the next slide. So this is some longitudinal plots that basically are making the point. Here what we're doing is that we're plotting the ADAS-Cog score as a function of time, and we're looking at the slopes for people who are amyloid negative or who are FDG negative on the bottom, and those who are amyloid positive or FDG positive on the top. So just look at those two plots on the right, one on the upper and one on the lower, and you can see the upper one where it says Florbetapir positive 0.9 points per year greater decline. That means that the people who had MCI, who had amyloid in the brain detected by Florbetapir, had a greater cognitive decline than people who did not have amyloid in the brain. And this is a theme that you're going to hear from me throughout and in the whole Alzheimer's field. If you have amyloid in your brain, you really have Alzheimer's disease pathology in your brain, and you are at much greater risk to show signs of cognitive decline. Here's another example of from the normal subjects. This is from 72 normal subjects. If you just look on the right, you'll see that the people, the lower right, the people that are Florbetapir negative, N at 49, their cognition is longitudinally is a flat horizontal line as a function of time. But if you look at the upper right, you see that their ADAS-Cog scores are going up. And in this case, a higher ADAS-Cog score means you have a problem. So the patients who have amyloid in their brain who are completely normal are showing a greater rate of cognitive decline than the normal people who don't have amyloid in their brain, suggesting that that amyloid is doing something that's not good and that these people are at future risk to have more decline and run into problems. The next slide here is from a different study. It was done in Australia. It's called ABLE. And it basically shows you the risk of having a positive amyloid scan. So if you look over on the right where it says MCI N of 87, you see that those people who are positive, there are 60 subjects there who are positive, and they converted 77% of the MCI patients who are amyloid positive converted to Alzheimer's disease in 3 years, where only 33% of the people who are negative converted to Alzheimer's disease in 3 years clinically. And if you look at the controls who are amyloid positive, 25% of them converted to MCI, which looked like early Alzheimer's disease in 3 years, whereas the negative, there was only one subject. So once again, a different study, but more evidence that having amyloid in your brain is associated with more rapid decline. Well, another way to assess the amount of Alzheimer's pathology in the brain is to do a lumbar puncture, otherwise known as a spinal tap, and measure the proteins A beta and tau in the brain. And you can see that if you go to the lower column where it says NC, if you look at A beta 1 to 42, it's 206. And then you can see if you look at the upper where it says AD, the A beta 1 to 42 has dropped to 143. So A beta goes down in the cerebrospinal fluid as A beta accumulates in plaques in the brain, and it's thought that perhaps the A beta is sticking to the plaques, which is why its concentrations are going down in the cerebrospinal fluid. In contrast to a reduction of A beta in CSF is the tau going up from 70 in the NC, which are the normal controls, to 122 in the A beta, in the Alzheimer's disease subjects. So we see a pattern here of CSF, cerebrospinal fluid, amyloid beta protein going down and tau proteins going up. And there are the measurements you get from CSF are quite concordant. They agree with the measures you get from amyloid scanning. So if you want to assess amyloid in the brain, you can use either PET scanning or cerebrospinal fluid A-beta measurements. The advantage of getting a PET scan is that you don't have to have a lumbar puncture. I mean, lumbar punctures are not a big deal. They're not particularly painful, but some people are reluctant to have them. So that's one reason to do a scan. You don't have to have a lumbar puncture, and it appears to be easier to standardize the scan measurements than the assays in the cerebrospinal fluid. But amyloid scans are costly, and you don't get any information about tau if you do a lumbar puncture. If you do a lumbar puncture, you can measure A-beta, you can measure tau, you can measure inflammatory proteins and other things. So you can get a lot of proteomic data from the CSF, and you can't get that from the amyloid PET scan. So the CSF provides many proteins, it's less expensive. There is this problem of resistance to lumbar puncture, and there are some problems with the immunoassays of CSF, A-beta, and tau, which make a standardization problematic. Using mass spec methods may be a help in this area. So this is some data from our lab just showing the hippocampal volume change over time in normal healthy subjects. And you can see that in just in normal people from ADNI, you can see all the raw data in all those colored lines. And the dark, solid black line shows you the mean and standard deviation of the rate of change of hippocampal volume over time. So, of course, we're all interested in the hippocampal volume does correlate with the memory decline. And we were interested in determining, well, what are the predictors or what are the various covariates that affect the rate of hippocampal atrophy. And you can see here that we looked at the effects of age, gender, intracranial volume, the gene ApoE4, the allele, which is a risk factor for Alzheimer's disease, white matter lesions measured on T1 scan, and amyloid measured from cerebrospinal fluid measurements. And you can see that the most significant predictor was amyloid, which had a p-value of 0.02. This shows you the rate of hippocampal atrophy as a function of age. The black line, these are all normal, healthy subjects. The black line shows the normal subjects who are amyloid negative, and the red line shows the normal subjects who are amyloid positive. And you can see that having amyloid is associated with increased rate of hippocampal atrophy. The red line is below the black line. But if you look on the scale on the left, you see that even people who are amyloid negative are showing considerable hippocampal atrophy over time, and it seems to increase with age. So there are factors that are causing brain atrophy that are not directly related to amyloid levels. Another way to look at all of this is to look at the different regions of the brain in people who are amyloid positive, which would be on the left, and the healthy controls who are amyloid negative on the right, to see if the regions are different. That is, does Alzheimer's pathology affect different regions of the brain than normal aging, or do we see similar patterns? And the answer, I think, is shown pretty well on this slide, that the regions that you see on the left-hand column, which are hippocampus, superior temporal, middle temporal, superior frontal, inferior parietal. And then if you go and you look over on the right, you see inferior temporal, middle temporal, lateral ventricle, fusiform, hippocampus. Those same regions on the right are the regions you see on the left. So what this means is that as people get older, there are regions of the brain which are shrinking, especially around the hippocampus and median temporal lobe, and the amyloid increases the rate of atrophy of these regions. And this all fits in with the idea that tau spreads into the region around the hippocampus and entorhinal cortex very early and is accelerated by Alzheimer's disease. This is a slide showing some diffusion tensor imaging results from Paul Thompson's laboratory. I don't have time to go into all the details. It's a complicated slide, but just to make the point that we did a lot of DTI and a lot of analysis has been done by Dr. Thompson, and that's available on the public website. This is more data showing longitudinal change of fractional anisotropy and diffusivity from the DTI data. And again, more data showing voxel-based analysis comparing the Alzheimer's disease subjects versus controls, showing the regions of the brain where DTI detects changes in patients with clinical Alzheimer's disease. And Dr. Thompson's laboratory has been mapping these group differences onto fiber paths and found that the measurements of mean diffusivity gave a better effect size, seemed to be more sensitive than looking at the fractional anisotropy, with the biggest changes being in the temporal lobe, which is, of course, where Alzheimer's disease is most prevalent. And here's some other connectivity graph analysis work of this. So there's a huge amount of very, very interesting various types of image processing and analyses that are done. Here's some the other work that's going on is arterial spin label perfusion MRI. This is a technique where the blood that flows into the brain is labeled in the MRI machine, and then a scan is done of the brain, and you can determine the rate of blood flow into the brain quite quantitatively by using this method. And here's a picture, for example, of a healthy control subject on the right, a patient with Alzheimer's disease on the left, showing reduced flow, especially in the back of the brain. Again, I'm trying to point an arrow to this and not able to, this is not somehow working. But at the bottom of the brain, you can see that the area is dark, reflecting reduced blood flow. And similarly, the MCI patient kind of is in between the control and the Alzheimer's patient. So here is a pretty complicated slide, but I think it's, and this data just came out in a paper by Nicholas Mattson, which is now published in Brain, just came out this week. But basically, what we're looking at here is we're looking at the effects of A-beta in normal subjects, in patients with early MCI, late MCI, and AD at the bottom. And we're looking at the effects of A-beta on the entorhinal cortex, hippocampus, inferior parietal. And you can see, for example, in the inferior temporal region, there is quite a substantial the effect of A-beta on the perfusion is a 0.25 effect size. That is 1/4 of a standard deviation of the perfusion was reduced in the amyloid positive subjects, whereas if you look at the volume effects, it's much smaller. It was only 0.04 of an effect size. If you look at the A-beta effect in early MCI, the next group down in yellow, in the inferior temporal cortex, you see that the effect in the ASL is 0.28, whereas the effect on volume was only 0.02. Well, then when you get down to the next group, the late MCI, we begin to see bigger changes in volume. For entorhinal cortex, this is the orange colored, if you go over to under the volume, you'll see 0.47 effect size, but if you go under ASL, it's only 0.14 effect size. So the big message is that in the early stages of the disease, A-beta seems to affect perfusion more than it affects volume. That is, it doesn't produce atrophy. But in the later stage of the disease, it affects atrophy greater than it affects perfusion. So there's a lot of questions that we're still asking. Which measurements best define people who are at risk for future cognitive decline and progression to dementia? Age is definitely a factor. It's the most important factor. Genetics clearly is a factor. People with family histories have greater rate of Alzheimer's disease, and they have a greater proportion of Alzheimer's disease. And the ApoE4 gene accounts for some of the risk of family history, but it doesn't account for all of the risk of family history. Cardiovascular risk factors also seem to be somewhat a risk for Alzheimer's, but it's not as less certain. Brain amyloid is definitely a risk factor, because if you have brain amyloid, you have Alzheimer's disease. Brain atrophy, in my view, is probably too nonspecific as a risk factor, but more work needs to be done. And then what is the role of ASL perfusion, DTI, resting state fMRI, and now we have tau imaging. So tau imaging is the kind of the hot new thing in our field. Despite the new scientific information and predictive value of amyloid PET scans, the truth is there is relatively poor correlation between amyloid PET and the clinical cognitive status. For example, as I've already mentioned, 30% of normal healthy people in their 70s have a high amyloid. Now, it's associated with more rapid cognitive decline, but you can have two people both age 75, one with dementia due to Alzheimer's disease, the other completely normal person, and they would have the same level of brain amyloid. So the correlation between amyloid and clinical status is not so good. But it is known from pathology that intracellular tau has a higher correlation with clinical status. So most of what we know about tau right now comes from pathology studies. And pathology, so here I've been instructed to click on the arrow again, and it should work for each slide, but it's not working. I'm doing something wrong, so I can't point. So I'll just describe the slide. What we know from pathology is kind of described here as the so-called Braak staging. Braak is a very, very famous pathologist in the Alzheimer's field. And what they're saying is, what Braak found is that people who are normal elderly have low levels of tau. That's called stages 1 and 2. But then in so I've been instructed now to use the whiteboard tools. I'm trying to use the whiteboard tools. I click on whiteboard, I'll click on it again, then I click on the arrow, and still nothing comes. So I'm definitely doing something wrong here, and I apologize that I can't use the arrow to click. Seemed to be working before. Anyway, the point is here that if you look in the middle of these two, if you look in the middle lower picture, you see a dark region on the brain, which describes the area around entorhinal cortex and hippocampus. And that's where tau first begins to collect in the very early stages of Alzheimer's disease. And that is why memory problems occur early in Alzheimer's disease. Alzheimer's disease is predominantly, not exclusively, a memory problem. And that's because the tau collects around the entorhinal cortex and hippocampus and associated structures. And then in later stages of Braak stage 5 and 6, the tau spreads all throughout the whole brain, and the person gets progressively demented. But the problem is that our knowledge now comes from pathology studies. Braak and many, many other people have written papers, but these are autopsy studies. The people are often not seen clinically, sometimes for more than a year before the autopsy. And of course, a lot of things happen often around the time of death, which influence the quality of the pathology. So our knowledge about the role of tau is limited, but there is a sense that it closely correlates with clinical symptomatology. So now there's been a lot of excitement in the field, and this is really what's hot right now in 2014, about measuring tau in living brain using PET ligands. And there are two PET ligand programs in humans. Well, there are more than two, but two that have been talked about a lot. One is a program out of Sendai, Japan at the Tokyo University, Professor Nakamura, called THK 523 and 5105. And then there's another group of compounds that were developed initially by Hartmut Kolb at Siemens, but now acquired by Avid, which is a pathology, which is a PET ligand company, which is now owned by Lilly, called T807 and T808. So I'm going to show you some of this data, but I just want to point out that the tau story is a complicated story. There are species of tau, so-called 3R and 4R. Tau is within cells, whereas amyloid is outside of cells. But even though they have all of these challenges, here's some examples of a tau PET scan from THK 5105. It was given to me by Professor Nakamura and Villemage. And you see that a patient with Alzheimer's disease who's in the lower side, there's more red there. You can see some red color, whereas in the healthy control above, you only see that red thing right in the middle of the brain. It's not clear exactly what that means. So there's clearly differences between people with Alzheimer's and healthy controls. And these investigators, again, showed that in the patient with Alzheimer's, you see on the right a lot more involvement of the brain than you see on the healthy control on the left. These investigators have shown that if you plot THK SUVR, that is the overall uptake of THK, against mini mental state exam or the clinical dementia rating scale, episodic memory or non-memory cognition, there's quite a good correlation that you can see on the top. So this is extremely encouraging that the amount of tau in the brain correlates with the degree of cognitive dysfunction. And here's another plot showing the relationship of THK 523 to hippocampal volume on the left. You can see there's a good correlation, but a very poor correlation with the amount of amyloid measured by PIB on the right. So now I'm going to show you some slides that come from the Avid company. This is the compound T807 developed by Hartmut Kolb originally at Siemens. And here's a group of subjects, normal control, going from the left, normal control, normal control, normal control, MCI, and AD. And you can see that the normal control subjects have very little tau, just maybe little blits, and there's some arrows pointing to them. But once you get to MCI and AD, you see a lot of red appearing in the regions where we would expect tau. So this is a kind of very exciting data. The patient on the left is a healthy control. The patient on the right is a patient with Alzheimer's disease with a mini mental state exam of 7. This is a very demented subject, but you can see there's appears to be extensive tau deposition. Another same case, the healthy control on the left, the patient with amyloid disease on the right. And you can see that it's much more yellow color, and you're seeing accumulation in the hippocampus area and in other parts of the brain as well. Here's 4 different subjects, a healthy control, a patient with mild MCI, a patient with mild Alzheimer's and severe Alzheimer's, and you can see that there's a progression of more and more tau in these subjects. These investigators also looked at the relationship of the tau uptake to the mini mental state exam, and you can see there's pretty good correlations here. Correlations of 0.9, 0.7, these are quite powerful. We don't see anything like this with amyloid. So the tau seems to relate closely to symptomatology. So therefore, in summary, we think that these early preliminary results, is now probably less than 200 patients worldwide have been studied, are very promising, and we can expect an explosion of new data. So we're very interested in what happens to people who are amyloid positive, and how much tau do we see in people who are amyloid negative. And what is the use of all of this in clinical trials? Would this be a sensitive outcome measure? Would the if a treatment slowed the progression of tau spread, would that be something that we could use as so-called a validated surrogate? A validated surrogate is something that everybody would like to see for clinical trials, because a validated surrogate would replace clinical outcomes. So you would not necessarily need to demonstrate that your treatment slowed cognition or function. You could simply show, if this was a validated surrogate, you could show that if your treatment slowed the progression of tau, the regulators and the scientific community would agree that you're really slowing the progression of Alzheimer's disease. So these are very interesting questions which we hope to be addressing in ADNI, because we're right now seeking funding to do tau PET scanning in the ADNI project. So changing gears a little bit here, we've been collecting DNA, and for those of you who have any interest in genetics, there's a tremendous amount of genetics information available in ADNI. Initially, we did what's called GWAS, and so the GWAS on every subject is available in the database. It's available right now. But in addition, thanks to the Wojcicki-Brin Foundation and the Alzheimer's Association, we were able to do whole genome sequencing of the DNA of 800 ADNI subjects. And the results have now been available. It's an old slide. The results have been available since last summer. It's the largest whole genome sequencing study done of Alzheimer's disease. And of course, all of the data is available to the community. So this is every single base pair sequenced in these 800 subjects. It's a treasure trove of data. And we're very excited about the results, and we haven't yet seen any papers coming out of this, but this is going to happen very soon. So another point to make about this project is the data sharing, that we, as I've emphasized before, all of the data that's collected in ADNI, that is all the raw data, all the process data, all the clinical data, the cognitive data, the imaging data, the GWAS data, the CSF amyloid and tau, et cetera, and proteomics. It's all there on the internet in a huge database that should be USC World ADNI, because Dr. Toga moved to USC. So I've got the URL wrong on this slide. And many people around the world have taken this data, written papers, and we have, well, right now, actually more than 500 manuscripts are now published, so that slide's a little outdated as well. And this unprecedented data sharing is really a model for future science. We would like to see more and more projects, especially big projects and small projects. Once people get their papers out, we'd like to see people sharing data on the internet so that other scientists can take advantage of the data, leading to more discovery and more rapid advances. Another thing I want to mention is something called the World Wide ADNI. The World Wide ADNI is, I've been only describing to you the results in the United States and Canada. We call that North American ADNI, and I showed you briefly some results from Australia. But there's ADNIs in Japan, Korea, China, Europe, and South America, and everybody is working together, and the data is being collected on the Aloni database. And it's a very exciting thing to create a worldwide community of investigators who are using the same methods and sharing data. This will greatly facilitate international clinical trials. I want to point out that ADNI is funded by the National Institute of Aging. And if you want to learn more about ADNI, you can go to our website, adni-info.org. That doesn't have the data. That's just an information site. It has all of the protocols. It has the grants that we wrote. It has the names of the investigators. It posts slides from our various meetings. So it's basically, if you want to get involved in learning more about ADNI, you could go to that website. There's also there have been 2 review articles that we published which review all of the papers in ADNI in a very, very thorough way. So you can, if you want to quickly see who's done what and what information has been gleaned, you can go to these review articles, and then if you get your own idea to do something new, that you can go to the ADNI database and analyze data and write a paper, and we love it. So these are the industry partners who are supporting about 30% of the funds to ADNI through something called the Foundation for NIH. We're greatly indebted to them for their support. And we have something called the private partner scientific board, which meets monthly to discuss the use of biomarkers for Alzheimer's clinical trials. So again, you can see these slides. I'll put all these slides up on the website, and you can go to the USC database to get the ADNI data. So, and then if you want to get your CME credits for this talk, you go to this site. And now we're at the last slide. So at this point, I'm going to go over and see who's asked for questions. I was told to click on this Q&A. So we have quite a large number of questions. Let me just get a sense of how many we have. Okay. So I'll just start taking the questions one by one as they come in, and then if I think we're probably going to have time to discuss these questions and even take some more. So if there's any of you have questions for me about ADNI or about Alzheimer's disease in general, or the use of imaging for neurodegenerative disease, I'd be happy to address them. So the first question is, have you ever seen the data on hippocampal volume and meditation practice in the alternative medicine field? And I think that this person is citing some specific paper, or maybe not. So the answer is yes. There's a whole world of data showing the effects of a lot of different things on hippocampal volume. Probably one of the most interesting studies that got this all started was some work that was done from England, where people showed that taxi drivers who studied for the London taxi exam had a bigger hippocampus than people who didn't study for the taxi exam. And since the hippocampus is involved with memory, this has been taken to mean that there's some plasticity going on, perhaps growth of new neurons or increase in size of neurons that is coupled to all of that memory work that people did to study for the exam. Since then, there have been many studies that have showed the benefits of many things on brain volumes, and exercise is one. Various types of cognitive training has been used. Various medications have been shown to cause change in various parts of the brain, and also have seen some work on meditation practice having a beneficial effect. So the next question I'm clicking on is, what is the role played by enzymes like inositol and triphosphate 3 kinase B in human brain and Alzheimer's disease? I don't know the answer to that. Inositol is not an enzyme. Inositol is a compound that's involved in signaling pathways. And myoinositol, which is one form of the sugar inositol, does seem to be elevated in Alzheimer's disease. But exactly what the role is of these enzymes is, this is not an area of my expertise. Sorry, I can't give you a better answer. So the next question is, what is the mechanism behind amyloid beta and tau protein proliferation? Let me click on this. I'm only seeing part of the question. It says, what is the mechanism behind amyloid beta and tau protein proliferation? Is it possible to use amino acid or enzymes to, but I can't see the rest of it, okay? So anyway, I'll answer, well, first of all, nobody knows the answer to the question between why we see a proliferation of these proteins. But we have part of the answer. There is a disorder called early onset familial Alzheimer's disease, which is a very strongly genetic form of Alzheimer's disease that affects families, and people develop in their early age. And so in those cases, there is a gene mutation that causes increased amyloid beta production. But in old people who get Alzheimer's disease, it's a little less certain whether or not there's increased production or perhaps decreased clearance of amyloid that results in the amyloid accumulating and amyloid plaques being formed. But there's a lot of interest in this, because if we understood the mechanism by why the amyloid went up, then we could develop treatments that were aimed at that mechanism. So that's an area of a lot of research. What is the mechanism between tau protein proliferation? Again, it's really not known. There's something about the interaction between amyloid and tau. Once people start getting amyloid in their brain, it causes tau to proliferate, but we don't know why. One thing that's been of great interest to people lately is the idea that these proteins, these are misfolded proteins, and that they seem to spread along the neuron and jump from one neuron to another neuron, almost in an infectious way, almost like a prion. And this is a lot of research is being done right now on what's called the cell-to-cell transmission of amyloid and tau. And some people are trying to develop treatments, including antibodies, that would block cell-to-cell transmission. So it's an area for a lot of research. It's not that an area where I have huge expertise. So is it the question, now I see the full question at the bottom, is it possible to use amino acid or enzymes to break down these proteins? I don't really know the answer to that. One of the major ways that people have been trying to get at the proteins is to use antibodies. And the antibodies can come from either what we call active immunization, that is using like a vaccine, and that was one of the really very famous observations that was made by Dale Schenk and published in Science 15 years ago, that if you inject amyloid protein into mice that are making amyloid in their brain, the antibodies that are generated in the mice will cause the plaques to go away. So there have been attempts to do clinical trials with vaccines. The first one, unfortunately, had a lot of side effects, and people were very badly hurt by it, so they had to discontinue the trial. Most of the approaches these days have been with monoclonal antibodies that are aimed at A-beta. But another approach is to use small molecules which inhibit the synthesis of A-beta. We call them secretase inhibitors because we know that there's a beta secretase and the gamma secretase which are involved in the synthesis of amyloid protein. So the next question is, I note that the controls in the tau PET ligand images are not age matched to the MCI and AD examples. Do these tau ligands PET intensity tend to increase in images with subjects' age? It's a very, very good question. The images that I've shown you are very, very early, a kind of proof of concept images. So I don't really know the answer to the question. We certainly expect, based on the pathology, that young people are going to have very little tau in their brain, and that as people get older, they're going to see more and more tau in the locus ceruleus, and the transentorhinal cortex, entorhinal cortex, hippocampus, temporal cortex. And as people develop amyloid in the brain and have Alzheimer's disease, there's going to be a much more rapid spread of tau throughout the rest of the brain. But we just don't know. Keith Johnson at the Massachusetts General Hospital is right now has done 40 or 50 studies, and everybody's very eager to see what his results look like. Avid Pharmaceutical is going to be distributing their tau PET ligand over the next months to some sites. So this is really hot. Everybody who's interested in imaging and clinical Alzheimer's disease is thinking and planning of how they can start doing tau PET scanning, because there's going to be a lot on this. It's certainly going to result in a lot of papers. We're going to learn a lot more about the pathophysiology of the disease. But more importantly, it's potentially a very good outcome measure for clinical trials. And that's because, in contrast to Alzheimer's, in contrast to amyloid beta, tau really correlates with symptoms. So if you slow the spread of tau, probably you're going to slow the spread of symptoms. That's what we would expect. And how you would slow the spread of tau? Well, perhaps drugs aimed at amyloid will slow the spread of tau, because certainly the presence of amyloid facilitates the spread of tau. But people are starting to develop treatments which are aimed specifically at tau. So it's just very, very exciting. The next slide is, what are my thoughts about autoantibodies as diagnostic markers of MCI or AD? Well, my thoughts are, this is a great thing to do. And there are quite a number of papers about autoantibodies going up in people with Alzheimer's disease. And the autoantibodies may be diagnostic markers. The autoantibodies might also be, depending on what they're autoantibodies to, they may be actually working to slow the progression or speed the progression of the disease. So I think it's a terrific target, and I think there have been some encouraging reports, but they, of course, they always need to be replicated. And that's the problem with all the blood tests involved in Alzheimer's disease. We have seen various papers get published where people claim to have good tests, but then it never gets replicated. So in science, you have to have replication and replication and replication so the finding can be ultimately generalized. The possible effects of DHA supplementation, omega-3s, fish oil, I would say that there are possible effects. There are a wide variety of stuff in the literature. I think that overall, I think it's fair to say that people who from very early life have been eating a lot of fish oil all their lives, they seem to have a lower incidence of Alzheimer's disease. But it's not so clear. You start taking fish oil as an adult, it's really going to reduce your risk for Alzheimer's disease. The epidemiology studies show associations, but that's not the same as doing a randomized clinical trial. And some small trials have been encouraging, but I think it's fair to say that right now the evidence is not strong enough that you could just routinely tell people they should be taking DHA. Although there are a lot of benefits claimed for other things for DHA, so it's associated with reduced cardiovascular risk and so forth. So, any idea why the hippocampus and entorhinal cortex are the first structures to be affected? The answer is basically no. We just don't know. There is some suggestion from Brock that tau originates in the locus ceruleus, and then there's a pathway from the locus ceruleus to the entorhinal cortex to the hippocampus, and tau goes along that pathway. But basically, in the whole field of neurodegenerative diseases, which we have Alzheimer's disease, of course, which affects memory and causes dementia, affected with amyloid and tau. Then you have Parkinson's disease, which is another neurodegenerative disease. That's associated with alpha-synuclein protein. That affects the substantia nigra and the basal ganglia and produces shaking and rigidity and difficulty walking. And then there's amyotrophic lateral sclerosis, which affects the motor system and causes paralysis and death. So there's Lewy body disease, which is associated with hallucinations and cognition problems. It's also caused by alpha-synuclein accumulation. But each of these disorders is associated with a different system. ALS affects the motor system. Alzheimer's disease affects the memory system. Parkinson's disease affects the motor system and balance. And we do not know why these proteins tend to collect in these specific networks or regions of the brain. So next question. Is Alzheimer's disease atrophy progression rate found related with any influencing factor? Well, people who are amyloid positive, people who have amyloid in their brain certainly have a more rapid rate of progression than people who don't have amyloid in their brain, even though the people are diagnosed clinically with Alzheimer's disease. So if we call Alzheimer's disease progression, the group of people who are diagnosed with Alzheimer's disease clinically, yes, it's clear that having amyloid in the brain is associated with a more rapid progression and brain atrophy. I think cerebrovascular disease has also been shown to increase the rate of brain atrophy, maybe the amount of white matter lesions, but I think that's more seen in normals and not so much seen in people with Alzheimer's dementia. I don't know if ApoE4 is associated with a more rapid progression. I just can't remember. I'm sure there are papers that are written on that. So I'm trying to think of what other influencing factors. There are age is an influencing factor. And I think that as people get older, like get into their 90s, the rate of change is slower. That is, in general, people know clinically It's really interesting about Alzheimer's disease. The younger you are when you get it, the more rapid the progression. If you get it in your 40s, it's a very rapid progression. If you get it in your 50s, it's still pretty fast. In 60s, it's slower. In 70s, it's slower. By the time people get into their 90s, if they have Alzheimer's disease, the rate of progression is very slow. Why older age is associated with a slower rate of progression is a big mystery. Next question. Astrocytes can be activated to help clean up tau tangles. It's a good idea. The danger is that if you activate the astrocytes or the microglia, they can produce adverse effects as well, because we know that in diseases like multiple sclerosis and HIV/AIDS, there is activation of the oligodendrocytes, and that is responsible for symptoms. So one has to be very careful about trying to develop approaches to clean out amyloid and clean out tau, because these approaches can have negative side effects. So I think that we're coming to the end of the questions as far as I can see. And we finished a little early. I think that usually people don't object to finishing early. So I think I thank you all for listening to the talk. I hope it's been helpful, and I wish you all lots of luck. Bye-bye.